Hey guys, so this video is on chromosomal mutations. It's the first section of our evolution topic and looking at what are the drivers of evolution. So our intention to this video is to know the different types of chromosomal mutations and to understand how mutations might then impact on evolution. So chromosomes, a bit of a refresher, they're the thread-like structures that are composed of our nucleic acids, so DNA or RNA, that contain our genetic information in the form of genes. So if we basically think about it, our chromosomes are really important to store our DNA. And if we actually break them down, they're just the series of bases formed into genes, which then form chromosomes, which are found inside our nucleus, inside a cell. So just to put that back in perspective. So chromosome mutations, and they're sometimes called block mutations, are where a section or a segment of that chromosome has mutated. And these usually lead to much larger scale changes in the organism that they've occurred in. And the ones we're going to look at today are inversions, deletions, duplications, and translocations. So you can see in this picture here are a wide range of chromosomes, and obviously they've gone through their different mutations. And I want you to imagine that each of these darkest bands represent a gene, let's say. So you can see that we're going to impact on multiple genes for each different mutation. So we'll start with inversions. So this is where part of a chromosome is removed and it's just replaced in the opposite order. So this normally doesn't have too harmful an impact, but it can sort of lead to errors in transcription translation. But generally, we've got all the genes that we need. So in these pictures here, you can imagine that letter A is a particular gene. So even after the mutation, we've still got the letter A. It's just that the other letters are in a bit of a different order. So we're still getting at least the genes that we require. Deletions, on the other hand, is where part of a chromosome is removed and it's not replaced. And so the impact of this is that those genes that were found in the section that had been removed are no longer present on this chromosome. And that could have detrimental effects on the organism. Again, it just depends on what genes have been removed. So in this example, it's the B gene that's been deleted from this chromosome. Duplications are when we've got a section that's been duplicated multiple times. And what this usually means is we've got twice the amount of instructions to make a particular protein, and we can get twice the amount of protein product. So again, depending on what gene it is that's duplicated, that could either be a really good thing or it could be a detrimental thing to the organism. So here you can see that we've got a double up of the gene B, which means we're going to have double the amount of protein eventually made by that gene. The last one we look at are translocations. So this is where two chromosomes, they're switched around. So there's different parts that are broken off, they get switched around and put back together. And this can sometimes disrupt like what is the order of the genes that we're expressing. It might even lead to things like cancer. But often a translocation has the biggest impact on the offspring, because if the genes aren't passed down correctly from a translocation, it means the offspring are then going to be missing certain sections as well. So chromosomal mutations, because they're much larger scale, I use them and identify them using a karyotype. And this is just a stained image of a chromosome under a microscope. So you can see here, for example, that part of this chromosome 7 has sort of been missing, but there's an extra part gained on chromosome 16. So this would be an example of a translocation from chromosome 7 to chromosome 16. So this is one way of maybe identifying some of those big block errors. An extra one that you could look at is if you had extra chromosomes. So Down syndrome, for example, you have 3 copies of chromosome 21, and that would be really identifiable in a karyotype like this. When we start looking at point mutations, you'll notice that a karyotype probably isn't that good to help identify any mutations. So in review, chromosome mutations impact chromosomes on a much larger scale, and they impact multiple genes at once. And examples that we've talked about are inversions, duplications, deletions, and translations. The next video we're going to look at are on point mutations, so down to single base mutations and what impact they have. So I hope this video helped.